We're now on top of the hour to start our lesson, which is lesson 10, titled Critical Review of Clinical Data for Efficacy. So before I continue, can I just have an indication if you can hear me? Colin?
Yes, we can.
Thank you very much. My apologies, my camera is not working today. So you'll be hearing my voice only. Please note that the lesson is recorded for future reference. Just looking at the agenda today, may I kindly ask that we mute and switch off the camera or the videos. You are most welcome to type in the chat any questions or comment pertaining to the lesson of today. As I indicated, for today, we are going to look at critically reviewing or assessing clinical data for safety and efficacy. And this particular lesson ties in very well with the ones that we've done. Interpretation of data, validity and acceptability of it for NRAs, and so on and so forth. So this will be presented by Professor Sam Salek, who is a professor in pharmacoepidemiology. And please look for the details in the platform. And followed by Prof. Sam, we will be listening to Kennedy, who will be giving us a lecture on hierarchy of evidence. Thereafter, we'll have a bio break, and then we'll break our way into our sessions as usual. Thereafter, we'll have a discussion on the groups, and then closure. So without wasting much time, I will hand over to Professor Sam Salek. Thank you, Prof.
Thank you very much indeed, Maps. That's a very kind introduction. Now I'm just going to introduce, without further ado, our speakers for this session. To begin with, the critical review of clinical efficacy is going to be introduced by, given by Dr. William Nickengoffak, who is a medical doctor, and a Cameroonian medical doctor, I should emphasize, an epidemiologist based at Northwest University in South Africa. Since joining the West University in 2020, his research is focused on patterns and determinants of medicine usage in South Africa in both public and private healthcare. And this is the bread and butter, those who work in the epidemiology space. It's a great pleasure. William is going to present the first talk and then followed by Kennedy. William, please.
Thank you very much, Sam, for the introduction. Just a sound check to please confirm if you can hear me clearly.
Yes, we can hear you.
We can hear you, William. Thank you.
Can you see my slide in presentation mode?
Yes, sir. We can see it. Thank you.
Awesome. Thank you. I'm just going to turn off my video as I go through the slides. Okay, so we're going to go through critical review of clinical data for efficacy and safety. And this is because being in the regulatory space, we have to do this activity, frequently look at evidence from submissions to make your decisions. So we'll have three main objectives for this session. The first will be to outline the steps of the evidence-based practice process. The second will be to explain the difference between what is a reporting checklist and what is a critical appraiser tool. And the last one will be to be able to critically appraise an article using the appropriate tool. So the evidence-based medicine was the initial term before evidence-based practice was coined. And initially, the term was developed because it was looking more of patient care by medical doctors. But due to the applicability of this approach to aspects of healthcare that are broader than medical care, whether it's nursing care, whether it's diagnostics, there's a whole variety of aspects of medical care that can be approached using this technique. It was now modified to evidence-based practice. So if you see the term, sometimes they're used interchangeably. And this term was initially coined by the late Dave Sackett, and he defined this as sorry, somebody speaking? Okay. So Sackett defined it as a conscientious, explicit, and judicious use of current-based evidence in making healthcare decisions. So it had to do with the way health practitioners make decisions, because initially lots of health decisions were made, based on just experience or anecdotal evidence, but it was an approach to help make things to be more rigorous and more, based on current practice or experience, such that whatever decisions are made can be verified against current practice and can be actually questionable. It was not a matter of people using their 10 or 20 years of experience to decide what was the best way forward. So it's now a mix, right? Evidence-based practice involves a mix. We all recognize that people have experience, and with experience come a certain amount of skill. But at the same time, people who have been practicing for many years need to be abreast of the recent development in terms of research or practice. So you have to merge the two, right? Integrating this individual experience with the base available updated research evidence. So there's 5 steps in this evidence-based practice. And these 5 steps follow a certain logical way. And the aim is to have a continuous improvement process. So it is a cycle, right, that goes from one study question to the next. So whenever clinicians are faced with a certain problem or a certain patient, and they have a challenge with addressing something relating to that patient's care, they have to first of all formulate a clear question. So formulating the clear question has to include certain key elements. And you probably read a bit more on those key elements in the references that are provided. But whenever a clinician or a healthcare professional is faced with a challenge, the first thing is to clearly define what that challenge is. You have to formulate a clear question. And there's different formats to that. It's supposed to have, like, who is the participant, what is the intervention, exposure you're looking at, is there a comparison? If there's one, what is it? And what are the outcomes you're looking at? So you need to define a clear question that describes whatever patient you have in front of you. Now, when the question is formulated clearly, the researcher or the practitioner has to go and find studies that answer the question. So you have to perform a search of the literature. There's certain criteria that have to be followed in order for a search to be rigorous. Let me put it that way. You can't just, search the library in your high school. There's databases, and there's trial registries where there are ongoing trials, and there's non-published literature, or what is referred to regularly as gray literature. Now, when this search has been completed, you find yourself with a certain number of articles or papers, or one. And having the article is not the end of the issue, because we know that clinical research or clinical papers have different sorts of quality. And when the quality differs, you have to look at the quality of that of the paper. So if a clinical trial is conducted, for example, and there's very poor quality of the trial, that it means that whatever the findings are have to be taken, into consideration of the quality. So that's why you have a critical appraisal step to look at this paper and the methodology and see if it ties with the results that are relevant to the patient whose question you are asking. Now, after assessing the methodological quality, you need to actually apply the results of this process to the particular patient who is in practice, or the question that you're looking at from a regulatory point of view. So it's not a theoretical aspect. You do all of this in order to prepare to make a certain decision. So you need to apply this to the question that you have before you. Now, when you've completed all of this process where you formulated the question, you've looked for studies to answer your question, you've appraised the studies to understand the quality of the evidence that you're getting to answer the study question you formulated, you've applied the findings of this to your case, your particular file or dossier or patient. The last step is to do some sort of introspection or some sort of review of the process to ask, how was this process done? Was the question properly formulated? Did you conduct a rigorous search? Did you really use the correct tool to appraise the evidence? And did you apply it well to the specific situation? So that last step is the audit process, where you review the entire evidence process and see if you can improve on it, or if there's lessons you could learn from it. Because being in the field, there will be the next file or dossier or patient that is met is a new problem, and you have to repeat this process over and over again. So when you repeat it over and over, you need to actually get better and be sure that whatever you're doing is actually optimal. So there's a reference there for you to look at, and that can be helpful. So like we talked before in the previous step, critical appraisal is the third step in the evidence-based process, and it has certain aspects to it. This includes looking at what you call the internal validity, which is looking at the methods that were done or that were used in the study, and see if these methods were actually appropriate. If you have a clinical trial and then there's no blinding of the intervention, then that's a problem, or if you do a clinical trial and you don't generate a random sequence to allocate your participants, then it means it's not a proper randomized control trial, which means there's consequences to the findings of that study. The next aspect is the reliability of the results. So you need to look at the results. What are the findings? Is there any difference in the groups that you're looking at? And if there's a difference, how precise is this result? Then finally, you have to look at the reliability of the results, the external validity. So external validity refers to the applicability of the result. So if you are in South Africa, for example, or you're in Kenya, and there's a trial that is conducted in maybe a low-income setting among children, do these findings apply to somebody who is practicing in Brazil? In a high income setting among adults. So, the findings of the results can only be applied to a restricted, number of people or variety of patients, because you cannot really extrapolate, you can't compare apples with pears, like they say, or apples with oranges. There is a resource which is also provided there, where you can have some introduction to critical appraisal. You can watch the video and learn a bit more and do a bit more reading around the topic. So there's a number of checklists that are used for critical appraisal, and this usually depend on the study design. So a number of organizations have developed certain checklists. I won't say that I'm listing them in order of, accuracy or quality. I'm just listing them in a totally random manner. When you use the different checklists, then you see which one meets your needs, and you can adopt whichever one you think is most appropriate. So there's the CASP tool, there's the tools designed by the Shaw unit, there's the Joanna Briggs Institute tool, and the other ones. Many of them have a lot of overlap in the items they look at and in the way they judge them, but these are individual tools that have differences, and you can use them where you see fit. So let us just pause for a moment and have a brief quiz. You can type the answer in the chat. or you can speak out. So which of these statements is false? There's two of them. I'll allow you a few seconds to read through, and then you can maybe speak out or type your answer in the chat. Sorry.
Yes, you can
Okay,
see
we're
the
already having
answers coming.
answers.
Mhm.
I can see the answers coming. Okay. We have
Most of them
25%
say
B
A.
and 75% A. Yeah,
Mhm.
yeah, yeah. Okay.
They keep coming.
They keep coming. I can see most of them are suggesting A as the answer.
Yeah.
So the option A says the steps of the evidence-based practice process are applicable only to clinical trials. And the second option says that the 5 steps of the evidence-based practice are ask, acquire, appraise, apply, and assess. Sorry for the typo. So has everybody responded?
They keep coming.
Okay.
Yes.
So let's just wait for 10 seconds for all the responses to come in.
All right. The majority is still A. Interesting.
Okay.
Yes, I
So
think now we can summarize it. Lillian?
yeah. So yeah. So the correct answer is actually B. Because the critical appraisal, sorry, the steps of the evidence-based practice. Remember I said if you have any research question that or dossier or any sort of patient in front of you, and that these steps can be applied not only by medical doctors, they can be applied in diagnostics, they can be applied by nurses, they can be applied by allied healthcare professionals, and they can be applied to any sort of question, which means it's not restricted only to clinical trials. Yes, they were initially developed to answer clinical trial questions, but they are not restricted only to clinical trials. However, the second option, the 5 steps of the evidence-based practice are actually ask, acquire, appraise, apply, and assess. I'm not sure if when I presented the slide, I used the word audit, and that's what maybe might have been a tricky part, but the correct option here, it's B. Okay. So now this tool gives you, or this slide gives you the tools and the study design for which they can be applied. We have 3 out of these 6 tools that can be applied irrespective of the study design. And this is really exhaustive. So you can use it for randomized control trials, cohort case control, cross-sectional studies, and even systematic reviews. However, there are some other tools that are more specific, like the British Medical Journal tool that can be applied only to RCTs and systematic reviews, and the Scottish Intercollegiate guidelines and the CAST tool. They can be applied to certain studies and not to others. So whenever you're working with these tools, you're going to get used to over time and decide which tool you think it's most appropriate for your use. So now let's move over from critical appraisal tools to reporting checklists. Many of you must have published papers in the past, and some journals, not all of them, do have requirements to say your article has to be aligned with certain reporting tools, reporting checklists. So the reason why you have a reporting checklist, or one of the reasons, is that there's discrepancy between what an author would say they did, so what they write in the actual paper, and what they actually did when they were conducting the study. So reporting checklists are actually made to get you to address certain aspects of the research methodology or the paper that will explain to the reader or will allow the reader to understand what is the link between what you're writing and what you actually did. So there are a number of standards that have been published, and this is actually just meant to ensure transparency of reporting. One of the most popular for randomized control trials is the consort statement. There's one for observational studies, the STROBE statements, and for systematic reviews is the Prisma tool. So if you're submitting an article to certain journals, many of the high impact journals or some of the journals that have good quality of outputs usually have these reporting tools that the authors have to apply to or have to adhere to. But it's good to always remember that these reporting tools are not critical appraisal tools. So it's not meant to look at the quality or to assess the quality. It's not a scoring tool. It's just a tool that allows the reader to actually make sure that you've adequately described critical aspects of your study. For example, most of the tools would look at the title and say, does the title actually reflect the content and does it include the study design? Because if you're looking for randomized control trials, if you're working in a regulatory file and the evidence is supposed to come from a randomized control trial, you don't want to open an article and download it and go and read the abstract and the entire methods to find out if it was a trial or not. That's just really a waste of time. So from the title, it should tell you if it's a randomized control trial. But you still don't end at that. Even if the title says it's a randomized control trial, when you're reading the paper, you need to look at the methods carefully to be sure that, it is actually a randomized control trial, because there's lots of papers out there written randomized control trial or systematic reviews, but when you read the content, it's actually not that design. So it's good to make a note here on what you call precision. You might address this when you're looking at the statistical aspects of the lecture. So whenever you estimate an effect of an intervention, there's what you call statistical significance. And when you're looking at, for example, a forest plot, this not only applies to systematic review. When you're looking at the results of a meta-analysis, you need to look at what you call the line of null effect, which means that it's an indicative line that tells you if there's a difference within the intervention between the groups that are being compared, whether they're comparing exposures or comparing interventions. So if you have one for the null effect, so one will refer to an effect estimate, which is a ratio. So something divided by the other, like a relative risk or odds ratio. Now, if the null effect is 0, it means it's for a difference. So different papers will report different measures of effect, and you just need to be conscious of the fact that the null effect is different. Now, you can have something which is statistically significant, which means that the two groups that you're comparing, they end up with a comparison p-value, which is, what it is. It is statistically significant. And in that case, the entire confidence interval of that output doesn't reach the line of no effect, right? So you will say that result is precise. For example, if you say the risk of developing high blood pressure if you drink sugar drinks is maybe 1.2 with a difference from 1.1 to 1.3, that is a ratio, right? And the difference would be like the difference in blood pressure between drug A and drug B would maybe be 3 millimeters of mercury. So that is a result that you have to look at. Now, that result can be statistically significant, but is it helpful for a clinician? So if a clinician is putting a patient on medicine for high blood pressure, does it really matter to the clinician if the patient is currently on a medicine that reduces the patient's blood pressure by 10 millimeters of mercury, and there's a new drug on the market that reduces the patient's blood pressure by 9 millimeters of mercury? So is that 1 millimeter of mercury difference sufficient for a clinician to say, I'm going to change the patient's treatment from A to B? So those are things that we have to look at, and it doesn't just depend on the p-values and statistical significance. So that is what we're referring to as precision. Now, there's other aspects of precision to say, is this actually, if it sees a difference, is that a clinically relevant difference? or if it doesn't see a difference, is it really that it doesn't see a difference, or the confidence intervals of that difference are really showing that it is there's no difference? Is somebody speaking?
Samuel,
Is
please
there a
mute
question?
yourself.
Okay, thank you. So I just thought it is important for you to know that there's some concept of precision in when you're looking at statistical results. There's also a new phenomenon, or new, but not really new, but it has become very popular in recent years, and is artificial intelligence and the application of artificial intelligence tools to healthcare research. Now, this is actually a threat because people have actually submitted papers to journals that have been published, and these papers have entirely been generated by AI, artificial intelligence. So knowing that several tasks can be done with artificial intelligence, you have to be able to distinguish what was generated by a machine and what was actually done by a human. And there are several tools for that, right? So people can do an entire literature review of an article or even a PhD thesis using artificial intelligence. Artificial intelligence can analyze data, it can write an article, it can extract data, it can assess risk of bias. So AI in itself is not a problem, but it is the application of the AI. So it's also worth noting that in general, there should be some human touch to say the AI is meant to assist a human do his job faster, not to replace a human. There are tools out there that do data extraction and risk of bias assessment that are AI-aided tools. Those are available out there, and it's not a bad thing on its own, as long as somebody can come and check to say, does this output make sense? Is there something really glaringly wrong in the way the AI dealt with the data? So these tools have been developed and is used in academia and even in industry. So there are other tools out there that are meant to detect AI content. So as regulators, you have to be aware of this, and you have to take the necessary steps to be sure that what you're looking at is really, it's not AI. So we'll just have a few minutes of another test. This one is asking, critical appraisal tools and reporting checklists can be used interchangeably. A, is this true, or B, is this false? We're just going to give 1 minute for people to post their answers.
Ask that everybody mutes themselves, please.
Are the answers coming in? How many answers do we have as of now?
Let's give it another 1 minute.
Okay.
There's more B than A.
Okay.
Okay, coming in. I think we can see that it's more B than A, Billion.
Awesome.
We
That
can
is
deliberate
exactly
on
true.
that. Yes.
Yeah, that's exactly so. A reporting checklist and critical appraisal tools are not interchangeable. They each have a specific use and application in medical research and in reviewing literature for efficacy and safety. So, in summary, evidence-based practice is a systematic approach that has 5 steps, and it provides guidance on how you assess, how you interpret, and how you apply evidence. Critical appraisal addresses internal validity, external validity, and reliability of a study. And study results interpretation of study results should not be limited only to statistical significance. Clinical significance should be considered when you are applying the findings of any study. This is some study material that will be sent to you, and you can look at it for further reading. Thank you.
Thank you very much, William. Thank you so much. I think we're doing fine with time. And now we will move to the polls. Am I correct, Steph?
We'll open for questions now, Maps, for the next 5 minutes.
Any questions from the faculty, from the students? Any comments? We have 5 minutes, or rather 3 minutes for the questions and answers.
A brief recap on your first question. I think there was some confusion about the answers because the question said what is false, and then actually the response A then should be false and B should be correct, right?
Lilian? You're muted. Lilian,
Maybe
you're
can
muted.
someone show
Can
the
you hear me?
question again just
Can you
to
hear me
avoid
now?
the confusion on the answer?
Can you hear me now?
That, would you like to summarize that?
Yes, Brian is absolutely correct. So the way I reported it was, could have been confusion. So the false question is A. So what is wrong is A. So if you said A is wrong, then you are right. I just wanted to avoid that negative way of saying it. I think I would adjust it again. So the statement A is false and statement B is true. So if you are asked this question, which of the statement is false, then you have to say A. So thank you very much for pointing that out.
Clarity. Okay, thank you so much. I saw a hand from Eunice. Eunice, do you still have a question?
Hi, thank you. That was actually the question that I had. Thank you.
All right, thank you. Thank you for that.
Can I ask another question, which is more relevant now for the regulatory assessors, and that is what usually is being done, or what these tools are being used for in clinical practice is for publications. And can you expand a bit more and explore the opportunity to use that also for regulatory submissions or clinical trial reports? With that, I think it could be certainly these tools are also very useful during the regulatory review process. But could you expand on that a bit more?
Is that a question for me or for the participants to explain how they use critical appraisal in their processes?
You can take that up, William.
Can you take that up? But I think it could be also for any comments from any of the regulators here in the session who have experience with this.
Yeah, I agree. I agree. So I'll allow any of the regulators who have reviewed study results to apply it in their decision making. I think it's better. Thanks.
Anyone from the team or the students? You're allowed to type in the chat. I think we can respond to that in the chat and then unpack that later on.
Yes, yes. I think the regulators sometimes review studies that were submitted, so they can be applied, but they can give more detail, like you say, in the chat or in the platform, and they can give examples of how they used it. So yeah, thank you.
Thank you, William. I think now we can go into into the polls.
Hello, MAPS. We're going to continue with Kennedy's presentation, but
Okay.
unfortunately, he's not with us as yet. He was traveling, and he was supposed to be stationed at this point of time and be able to join. What we are going to do, I'm going to make an attempt to give his presentation, and of course hand over to him, when he joins us. So the title of the talk is Distinguish Clinical Significance from Statistical Significance. Now, this particular topic has been a debate for many years among the researchers as well as the regulators, how to distinguish between clinical significance and clinical significance, and whether when we do have a statistical significance, we should base our decision just on that, or we need to have some sort of a balance between the statistical as well as the clinical significance. So that is really the main thrust of this, and we hope that by the end of this talk, and hopefully debating this further in the breakout rooms, there will be a clearer understanding of how we should implement, and when and how we should put more emphasis on the statistical, and when on clinical, and when on the combination of the two. And of course the pros and cons and the pitfalls of every one of those, if we do look at them in isolation. So the objective or the learning outcomes of the lecture is to highlight the importance of understanding those concepts to make informed regulatory decision. So in terms of what you would take away from this talk, is really a greater understanding and application of the clinical and statistical significance in your decision making. The second objective is overview of relevance in regulatory setting, and being able to explain how regulatory authorities often need to balance the statistical outcomes with real-world clinically meaningful effects for patients. The more and more, and I'm sure that as regulators you already know that greater emphasis is placed on the real-world evidence, and the real-world studies becoming and gaining more impetus, and in some instances of a regulatory process, when it comes in particular with the drugs for unmet need, that they go through the fast track or facilitated regulatory pathway, they often required as the post-licensing commitment or post-approval commitment that the companies would also submit real-world data. And that's on the strength of, because at that time with the limited approval, the drugs get marketed, and therefore that exposure of the society or the greater number in the society to that drug would be of benefit to the regulators to see whether they want to change that temporary or limited approval to a permanent approval. Next slide, please. So why this is important, I'm sure that you all know why this particular topic is important to understand in your role as regulators. I suppose the underpinning concept for this is that in a large majority, I can't think of any studies that you evaluate that would not be a hypothesis testing studies. And one of the other things that you would be looking at to see whether the statistical test that is being applied is the correct one. Say, for example, what is the important test to use? when you're looking at the superiority trial, when you're looking at the inferiority trials are different. And if you're looking at the inferiority trials, you know, what is really the statistical test to use to show that margin of difference? So, so that is really the key, and the components of that hypothesis testing are really the power of the study, and in terms of the, you know, how we evaluate the result, you know, whether, you know, they have looked at the, you know, the type 1 error and the type 2 error as part of that complete package of the statistical policy, or the statistical, or often be referred to as SAP, the statistical analysis framework or policy. Next slide, please. And the other situations are that sometimes finding presented can be difficult to interpret, and, you know, whether, you know, say, for example, in recent years, you know, we've been looking at the, you know, patient-reported outcomes, and not everybody in a regulatory authority as regulatory professionals would have enough experience in, you know, looking at those data for, you know, patient-reported outcomes. And for example, you know, if you look at the, you know, the NRAs in Africa, majority of them, you know, do not include the benefit risk as part of the assessment template. And that sometimes, you know, could be challenging, you know, how to interpret and how to make the right decision. So therefore, you know, it becomes, you know, critical that the regulatory authorities that be able to interpret the clinical and statistical significance of the clinical trial in the right context. And to be able to, following that, to be able to make judgment on whether statistically significant results are clinically important too, and how we can actually manage that tension between the statistical as well as the clinical significance. Next slide, please. Next slide, please. Thank you.
Prof, I just need to bring to your attention that Kennedy is in the room now. I don't know
Okay,
if
well,
you'd
I'd
like
be
to
more
hand
than
over
happy to hand over to Kennedy. Yes.
Thank you. Thank you very much for that good introduction.
I was getting to the more interesting stuff than I was getting excited. Here we are.
Okay.
Kennedy, over to you.
Kennedy, over to you, and welcome.
Hi, everyone. I'm sorry I'm a bit late. The weather and many things outside of my control, but I'm here now.
So Kennedy, welcome, and I'm glad that you arrived, that you're based safe and sound. Could I just you know, give you a short introduction before you start? And I'm sure that you don't need introduction among the audience that we have, but perhaps one or two may not know you. Now, Dr. Kennedy is a professor of medical statistics, and he wears several hats, and I will just suffice to you know, tell you two of those hats. He is with the University of Witwatersrand in Johannesburg as a professor of medical statistics with a specific remit on the prenatal HIV research with the Faculty of the Health Sciences. And his second academic hat that he wears is with the School of Public Health Faculty of Health Sciences, and again with the Witwatersrand Johannesburg University. It's a pleasure to have you with us, Kennedy. Over to you.
Thank you. Thank you. Hi, everybody. Those who were in my previous meeting would remember me. I work as a biostatistician, as you have been informed. I happen to be all over, and today I'm taking this call from outside of my usual space. So what is clinical significance? So I first want to thank Sam, Stephanie, Colin, and all the others who've come in to you know, take my place as I was trying to connect. I see even Villian too was online. So let's talk about clinical significance. So what is clinical significance and what does it assess? So it assesses whether differences observed in a clinical trial have meaningful impact on patients. So Oftentimes, as regulatory officials, you will get data, you'll get dossier that has information about how a study performed, and you have to make a decision on whether the product needs to be registered or not. And you look at what impact or what effect was seen in the study that was done. And so you tend to look for effects that touch on health, improvement of quality of life or functional status. Hence, the key thing you're actually looking at as a statistician at that point in time, you are reviewing, but you wear the lenses of a statistician is there some minimally clinically important difference? I'll just call this MCID for now. So this is a very important point, and those of you who engage in clinical research would understand that as you're designing a study and you want your sample size done, the first thing a statistician will ask you is what is the minimally clinically important difference you want to achieve with whatever intervention you want to test against a placebo arm. And from that conversation, then the statistician is able to move on and give some numbers based on your guidance in terms of the minimally clinical important difference. However, you as a regulatory officer then has to make a call between what this means in relationship to statistical significance. So ideally, you want something that has an impact on patients' health, quality of life, and functional status. Let's go to the next slide. So, but it's not unusual that you find that you've got a wider effect size. So we don't just look at the p-value. You look at the p-value and then you look at the effect size. When we say the effect size, we're talking about the 95% confidence interval. So some of you would have reviewed those years, which is that it comes with an odds ratio value with 95% confidence interval or hazard ratio or relative risk or any other assessment, but those are the most common ones. And ideally, the smaller the sample size as you are doing your sample size calculation, so if you have a very big effect size in your sample size calculation, then you're going to be looking at a smaller sample size. And if you have a narrow effect size, then your sample size is going to be. And a lot of these things depend on many other things, logistics, how much you have available to do the study, what is feasible logistically. So all those things play a role in terms of what you settle down for as a sample size. So you as a regulatory official that is reviewing documentation, your interest should then lie in terms of the minimum effect size. What is it that you can see or you have seen in this application that is beneficial or that is likely to be beneficial? So that's the important question you need to ask yourself in your review. And the good thing is, or the bad thing is, that question is a statistical question. So it's not only a clinical question, it has statistical implications. Let's go to the next slide, please. So I had given an example over there. There's an example you've just jumped. The slide just before this. If a new arthritis drug results in a reduction of joint pain by an amount that enables patients to resume daily activities or reduces the need for other pain medication, so this change would likely be considered clinically significant because you can see this positive change in the participants. So theoretically, this is what you want to hear. This is what you want to see. But now let's look at statistical significance and then we will make a call. Then you have this thing called statistical significance, often assessed by a value called P that is less than 0.05. So your statistical significance is basically a value that is estimated using some mathematical approach. And what that does is that it helps you determine if an observed effect or difference in a study is unlikely to have occurred by chance alone. And then we look at the p-value. The p-value usually has a threshold of 0.05. When you're doing your phase 3 studies, we work with 0.05. But when you're doing your earlier studies, like maybe a phase 2 or phase 1, you potentially want to have a lower chance of error. So you wouldn't generally work with 0.05. You would go down to 0.025. But oftentimes the conventional value is 0.05. So it's that value that we tend to use once you've identified your hypothesis. And then when you get your results, then you make a call whether the hypothesis has been rejected or not rejected, basically based on the statistical significant value. Now we get p-values often, but you can also calculate this by hand and get what is called your statistical value, and then you look at the statistical value against a critical value, and then you make a call on whether that value is significant or not. So this whole conversation of whether the estimation that you have in front of you is significant or not depends on the definition of what is called a hypothesis. You usually have your null and the alternative hypothesis. So I won't go into the details of that because you already know that. And then based on the p-value that you get, or the your statistical value compared to the critical value, then you can make a call on whether there is some significance or not. Let's go to the next slide. Now, for example, if a drug trial shows a statistical significant reduction in blood pressure with a p-value below 0.05, but then the effect size is a reduction of only 1 mmM per Hg. So this might be statistically significant, but not necessarily clinically significant. Now, this is where your judgment as a reviewer comes in handy. What does this mean? Do you just take this product and then go out and register it, or accept that it be registered because it has a p-value that is significant? But you also have to look at what reduction are you seeing, and is it worth it? I'll explain why some of these things happen because of statistics, and then you'll see the point why we are here. So let's go to the next. So again, your statistical significance is based on what we call a type 1 or type 2 error. So type 1 error is basically a false positive rate, which you would know, so I won't go into the details. But in simple terms, without even going to the depth of statistics or mathematics, this occurs when a clinical trial incorrectly concludes that a treatment is effective when it actually is not. So basically, it just says it detects whether an effect is not present. And then, of course, a type 2 error is what we call a false negative. And that does occur when a clinical trial fails to detect an effect that is actually there, concluding that a treatment is ineffective when it actually has a beneficial effect. So these are the real issues as a regulatory reviewer that you are going to face, and you have to make decisions. And based on what has been put in front of you, you would need to apply your mind, looking both at the clinical significance, the statistical significance. But statistical significance, don't just look at the p-value. You also want to look at what is the effect size, and then understanding that making a wrong decision around that will either land you in a type 1 error situation or a type 2 error situation. So you also want to weigh the decision or the conclusion that you make to avoid getting into any one of these errors. And in fact, I'll pose a question to you. You can answer this in your groups. Would you prefer, or rather, which one is bad? Which one is worse? Is it a type 1 error or a type 2 error? Okay, let's go to the next. And this is just basically a Bell distribution showing you theoretically from a mathematical point of view where this lies. And you have an upper tail and a lower tail. Usually your alpha is 0.05. Then when you divide it by 2, it is 0.025 on the top side of the tail and 0.025 at the bottom. If you're in the middle, that is a region we are saying do not reject. That is where 95% of the time you're in the safe region. You don't reject the null hypothesis. But once you cross the critical value, which statistically, it could be any other value that you've set, but conventionally we use 1.96 for 0.05 divided by 2. And if you fall in either of those red regions, then those are the regions theoretically we say that, well, you got to reject your null hypothesis because of it landing in the regions that are shaded in red. So theoretically, this is how statisticians think, and this is how they make a decision. Let's go to the next slide. But what are the limitations of statistical significance? So this is what I was alluding to. So one must remember that statistical significance is heavily tied to sample size. So if you have a small sample size, you're very unlikely to detect a true effect if it actually exists, because a small sample size has less power to detect differences, and therefore increases the risk of the risk of landing in a type 2 error, which basically is failing to detect an effect that is present. And then also remember, under limitation of statistical significance, that it also has an effect on p-values. Now, and this is where, again, as a regulatory official, you got to be very careful. Now, when you do a clinical trial that has very large sample sizes, remember, large sample sizes can show a significant difference, even when there is a very small difference between groups, which we will say it's clinically insignificant. And that then lands into what's called a type 1 error, that is detecting an effect that is actually not present. But this difference is a difference that you're seeing because your sample sizes are very, very large, which is the reason why before you engage in a study, especially a clinical trial, it's very important that you apply your mind in terms of the statistical aspects of study design, sample size, how you're going to do your recruitment, your sampling frame, your randomization. Because if you make a mistake with this, if your study design is poor, it's very easy to land into any one of these challenges, which would then set you in a position where you may end up making a type 1 or a type 2 error. And in essence, what that means is you may end up rejecting an application that comes to your desk because you think that it is not effective, when in fact, it may have been effective. It may have been effective, it's just that it was designed poorly. Or alternatively, you may also register a product that is actually one that did actually not find an effect. And in essence, as a regulatory official, you would then be exposing people to a medication that has not been well studied, and it may as well actually have a negative effect. So these are critical decisions that, as a regulatory official, one needs to apply their mind as you are reviewing applications sent to you. Let's go to the next slide. And then also remember, I did talk about effect sizes. And if you have a small sample size, the problem that you will almost immediately face is that when you're looking at effect sizes, so effect sizes is just simply the 95% confidence intervals. If your sample size is small, then you're going to have wider intervals. And if your sample size is sufficient, you will have narrow confidence intervals. So one other way I would urge you as a regulatory official, when you're reviewing dossiers that are brought to you, is don't just look at odds ratios and hazard ratios and relative risks on their own. Look at those together with 95 confidence intervals. And if you find that estimates reported have wide intervals, ask questions. Why are those intervals wide? And then that might give you an idea, because oftentimes an interval is wide because there is sparsity of information, and or there is missing information, and for that reason you find a wide interval. I was once in a meeting where somebody presented a risk of death in under 5 mortality in one of the countries in Africa, and he found a significant odds ratio. I think the lower value was about 2.5, and the upper value was more than 1,000. And he was very happy because he had a p-value that was significant. But then I asked him, I wouldn't mention names or the institution, but then I asked him, do you understand what the confidence interval that you're showing there means? And that is when the reality hit him. And he had wanted to argue. Then I said, look, let's just make it easy. Let's look at your data. And when we went to the data, we found that it was actually true that he had a problem in terms of the numbers, and there was a lot of missing information, which then pushed out the confidence interval. So this is a knowledge that you roughly need to have. You don't need to know the technical details of it, but understand that if the interval is wide, then it has a meaning, and that meaning is what you want to look at and ask yourself whether that meaning will allow you to register a product that is on your desk that's been submitted for approval or for review. So always have these thoughts in your minds when you're reviewing a dossier or maybe even a manuscript. And then, of course, ask those who are submitting the documentation what they think that means, and then raise your fears so that they can get to understand why you have some level of trepidation regarding the application that they put forward to your desk for review and approval. Let's go to the next slide. Then there is a precision of the estimate, which is basically the effect on p-values. Although I've talked about this, but just remember that very large sample sizes will give you a significant p-value, and that significant p-value may not necessarily be clinically significant. So again, make your judgment based on that. Just because the p-value is significant does not motivate you to just say, yes, I'm going to go ahead and approve this application. You also need to look at the effect size. Is it worth doing the registration? Has this study been, tested, say, in other populations? And have you seen a variation in that effect size before you make that call? So precision is important. And it is something that you also want to understand as a regulatory reviewer and make a proper call when making a final decision on application sent to you or some application submitted to your desk. Next. So I'll give you a simple example here. So you have a study whose objective is to assess systolic blood pressure. It plans to enroll 5,000 participants, or actually has enrolled 5,000 participants, 2,500 per arm. One group is the treatment group, and the other group is the intervention group. Sorry, it should have been placebo. Sorry, it should be treatment and placebo. Follow-up is 3 months, and the primary outcome is change in the systolic blood pressure. What you're interested in is the average reduction in the intervention arm. And you find that the reduction in the intervention arm was 2.5, and in the control, it is 2, showing that the difference is 2.5 minus 2, which is 0.5, but it's a significant p-value. Now, the question you want to ask yourself is that, well, this is statistically significant. I mean, clearly, the sample sizes enrolling in that study are huge at 5,000, but what is the effect size? Would you approve a product that's been submitted to your desk for review if you see an effect size of only 0.5 in patient health that is likely too small to be relevant? This is a reality that you face as a reviewer when documentation for review comes to your desk. So bear in mind that when the sample size is huge, again, ask yourself, is that significant p-value a spurious one? And should I go ahead and just approve this product because of such a small benefit? Let's go to the next slide. So this would be the MCQ. And the question here is, why is it essential for regulatory authority staff to interpret clinical and statistical significance accurately? You've got option 1, they need to ensure all clinical trials are conducted by the same research team. B, they are responsible for assessing the reproducibility and interpretability of trial findings. C, they aim to prioritize trials with statistically significant results only. And D, they are primarily focused on reducing the number of submissions. What is your response?
We're getting some answers coming.
Oh, awesome. That's a correct answer,
Yes.
B. Yeah.
B.
They are responsible for assessing the reproducibility and interpretability of trial finding. Correct? Nice, 95%. That's amazing. Yes, it's B. 2, what challenge do regulatory authorities face when reviewing clinical trial results? 1, all studies are easily reproducible and straightforward to interpret. 2, clinical trials rarely test hypotheses as part of evidence-based medicine. 3, some studies present findings that are difficult to interpret or are not reproducible. And D, regulatory authorities do not focus on statistical or clinical significance. What challenge? I'm very curious to see what responses you give here. Amazing. C. Yeah, some studies present findings that are difficult to, to interpret or not reproducible. Amazing. I'm glad we are generally on the same page. So what is your role as a regulatory authority official? So what you have to do is balance the evidence presented to you. Assess the statistical evidence that is right on your table in the documents that have been submitted to you for review. Ask yourself, is the sample size sufficient? Ask yourself, did they report on the power that they used to calculate the sample size? And do you think that was sufficient? You may or may not have the statistical capacity to make that knowledge, but there's always no harm in consulting a statistician or somebody that has an understanding of that. Then, of course, there is precision. You assess precision. Look at the confidence bands. Are they narrow? Are they wide? If they are wide, throw harder questions and ask the applicants, do you know what this means? Because there is doubt here that, you seem to, but having said that, also remember that you may also be dealing with some of these rare studies that cannot have the luxury of enrolling huge sample sizes. Again, this is the reason why we're talking about balancing evidence. Some studies, say in our own setting, in Southern Africa, or let's say in Sub-Saharan Africa, HIV is big. So usually the studies we do in general would have sufficient numbers. But in some other studies, like some specific cancer type studies, you'll never have a huge sample size. With that comes the problem of precision. So this is where you apply your mind as a regulatory authority and balance what is in front of you by assessing the evidence, what can be done logistically, and you make a call. And then, of course, the second bit as a regulatory official is, there's often guidelines for decision making. Some of you in your regulatory authorities, I just cited a few here, SAPRA PPB and the Ghana FDA, but there's many others here from Zimbabwe, I know as well, and Nigeria. You have guidelines that help you make a decision in terms of what to assess clinically and significantly. So those structured frameworks are quite helpful in trying to ensure that there's some level of uniformity that one follows in making a decision. You don't want to make decisions that you can't justify. So it helps balance decisions that are made, because if you stick to the structures proposed or the structures laid out or the framework laid out, then you're able to make a call knowing clearly that at least you've used a framework that's been put in place to help you make that call. And of course, as I've mentioned, those ones do help in ensuring that there's some level of systematic process and transparency in the decisions you're making in terms of final decisions on applications sent to you for assessment. Let's go to the next. And then there's also the risk-benefit analysis. Those of you who review work that touches on data safety and monitoring board, this risk-benefit analysis is a big thing. You want to make sure that you weigh interventions that are being tested against what placebo arms are being given to ensure that there is no harm. So you as a regulatory person, as you are reviewing those years, yours is also to ensure that, you know, potential health benefits versus risk are looked at carefully so that you don't want to cause harm by approving a product that's not been properly assessed. And this is where you need to also apply your mind in balancing efficacy and safety. Efficacy and safety is what we generally look for in trials. Any statistician will tell you efficacy and safety. While we're interested in efficacy and showing that the p-value is significant, the bottom line and the most important part of a study usually is the safety component, especially when the drug is new and getting into the market, and you know, you want to be sure that it is safe and won't cause harm. Safety is critical. And of course, there's also guidelines in terms of what you need to look at, what you need to look for, what grading of conditions you need to look at, and whether those are SAEs, and you know, how quickly they've been reported, how well you managed it. And so that risk-benefit analysis is also transferred to you as a reviewer when making a decision on an application sent to you for review. Then, of course, there's also the patient-centric decision-making. Now, this is interesting. This is where you try to incorporate patient-reported outcomes. I can tell you it's not been very easy. I'm involved in trials. It's not been very easy, but you know, we are winning. One of the studies that has done pretty well in terms of engaging patient-reported outcomes is studies that follow what we call discrete choice experiments. This is where you first talk to potential patients or participants to ask them, say, for example, if PrEP is made available, would you take PrEP that gives you some negative effects like a headache, or PrEP that would lead you to vomiting if you take it, or PrEP that would require you to, you know, take the medication regularly, or you just take a jab? Now, when they give you that feedback, and then you take that feedback and incorporate it in designing your study and incorporating their thoughts, then you're doing what is called a patient-centric process in designing your study. So it is usually useful where you can. I must be honest with you, in some studies, actually in many studies, it doesn't happen, but in some studies, it has happened very well. I can almost guarantee you that a lot of the PrEP studies have done that very well, and that's worked very well. I've also seen some of the studies that SAM has done in the UK. They also have done a lot of patient-reported outcomes. You know, they have involved patients in terms of designing studies, in terms of also talking to them to interpret the findings. So that's the other way of looking at it. And then, of course, you can also use real-world evidence. This is pulling information from registries to try and help you understand, why is a p-value significant? Why is the confidence interval tight or wide? So those registries are very important. And if I'm not wrong, I think I've come to the end of the talk.
Was I being subtle, Kennedy?
Yeah, no, your timing was good.
Okay. Back to you, Maps.
Thank you, Colin. We will now have about 5 minutes for questions. And then soon after, we'll have a bio break for 5 minutes. Then soon after, the breakaway rooms. Any questions from the floor?
If the floor doesn't have questions, I'm asking them a question. What would you think is less harmful, a type 1 error or a type 2 error? I don't have an answer. I just want to hear from your experiences when you are reviewing documentation. What do you think?
Kennedy, while you're thinking, I just want to add a comment that might help them. I think, you know, in order to remember, first of all, the importance and how distinguish between the two, you always need to think, which one of the two is the best decision, sending the innocent man to jail or freeing a guilty man from the jail? So that type 1 and type 2 error is really, superimposes to that sort of decision making. And that's exactly what it is, as you described in your talk. Setting the guilty man free or putting the innocent man to jail.
Okay. I think Kennedy will type that in whatever response he has in the chat. Can I ask those who have their hands up to type in the questions in the chat? We will try and respond to them later because we are running a little bit behind time. And I will allocate 3 minutes for bio break. And then soon after that, we'll have the breakaway rooms. 3 minutes. Thank you.
Thank you, Maps.
Okay, so we're going to break into our rooms for the breakout sessions. And Stephanie will take us to the rooms. You have seen the questions. And can I urge the members to please immediately choose the rapporteur? Thank you, Steph.
Hello.
Hi, Prof, you're in the room.
I'll be back in the room. Lovely.
Yeah.
That's great. So are we going to go first, Stephanie?
So yes, you're welcome to moderate the sessions from the odd numbers all the way through to the even number rooms for the questions that are listed.
Sure. I think we're going to go to odd numbers, and if I could call upon Finio to as the rapporteur to present the deliberation in 5 minutes, please. Not more than that.
Thank you to everyone. Hopefully everyone managed to finish the 2 questions that were given. Very interesting interaction that we had in the group. I think I will hand over to the groups.
Thank you, Maps.
Over to you, Sam.
Thank you. Now we're going to take the odd numbered rooms. Could I have Finio, please, as the rapporteur? Hello? Finio, are you there?
Yes.
Okay, lovely. Great. Take it away, please.
All right. So,
Are you going to present the slides?
no, I think I'll just.
Okay,
I'll
lovely.
just
Carry on, please.
speak from there. So we are considering the odd numbered groups, question 1 and 2. So we had established that it is critical to consider both significances, that is the statistical significance, as well as the clinical significance, as we believe they are important in decision making. More
Could I ask,
especially,
please, put yourself on mute, please? Carry on, Finio, please.
yes, as we believe they are important when making informed decisions about drug approvals. And then they are also, when you look at the clinical endpoints, we established that even if a product maybe is displaying statistical significance, you also have to follow up maybe the clinical endpoints to see if, even despite its positive statistical significance, are you also getting the positive significance from a clinical standpoint. And then we also talked about how sample size also affects the significances, and therefore it's critical that the regulators are aware of this. Like when you have a small sample size, for example, it will give you a wider confidence interval, whereas when you have a larger sample size, the confidence interval becomes narrower. So that then speaks to you having to also look at the procedures, the processes that were performed, the sample size. That is, in essence, look at the methods. And then you can also maybe check your systematic review and meta-analysis, whether those were performed as they can also help you establish the significance of the study. So I think those are the key points that I have captured for the group.
Lovely. Thank you very much indeed, Finio. Now, any questions from the floor regarding the topics of the odd-numbered room? Or Kennedy, did you want to add to that? Any comments?
No, I think he's presented it well, but what I wanted to mention is that and I won't allude to this, but the conversation around regulatory authorities, or rather regulatory authority officials understanding the difference between statistical significance and clinical significance is important because they make heavy decisions, and they need to weigh that decision carefully because you don't want to put a drug in the market, yet it can cause harm. It's yet to be well understood. So it's so important to have a good understanding of this and understand how best to make that final decision, and always consult. Don't do a one-man decision. It's always good to consult and get the opinion of other experts in the field before you make the final call. Thanks.
And there are, as you know, Kennedy, there are some really unfortunate experiences in the past, and the regulatory authorities have been taken to task, for making that wrong decision. For example, in the case of the major one is really the case of Vioxx in America, that FDA was taken to task by the Congress, and that brought quite a number of issues for the FDA, going forward from that particular point. And in fact, that was the turning point that quite a lot of the dinosaurs in the FDA changed, and the new management were brought for different sections, PDUFA and others. So Kennedy, who did you have as a rapporteur for the even number?
Yeah, so we had Jessica.
Okay, so that's for group 2, right?
Yes.
Okay, so question was discussed why reproducibility of trial results is critical for regulatory approvals. It's very important because it's the duty of regulatory agencies to approve medications that are not just safe and of good quality, but medications or devices or whatever it is that are efficacious. So if a drug has proven efficacious in a clinical trial setting and this drug is to be approved, the medication or whatever we want to approve should have the ability to produce the same effect across the population. If the trial setting, we're able to get a particular response from the trial setting, but we didn't get it across the population, it's going to be a problem in the regulatory work. And also, if the reproducibility fails, there's going to be a lot of issues for the regulatory agency aside the health implications on the population. We are going to have to send out communiques to do a recall, and even with that, it's not, we don't always get all the products being recalled. Some people may not even hear about it, and they may still be using the medication. So it's very, very important for us to have reproducibility so that we don't have this back and forth when it comes to a failure in the efficacy of the medication. And for the second question, describe how regulatory authorities evaluate if statistically significant findings are also clinically important. So for this point, it was quite interesting because for most of the agencies, we were more concerned about the clinical aspect, and not questions, but ideas were coming about the fact that, I'm so sorry about the background noise. I'm really, really sorry. Is it better?
Yeah, it's better now.
Is it better? Okay, thank you. Okay, so for how we evaluate, even in Prof. Kennedy's presentation, he mentioned that not all statistically significant findings are clinically important. And for us, we dwell more on the clinical importance, even as we look out for the clinical significance. So we look out for our p-value, we look out for the, the confidence interval and all that. But even with that, we are more concerned about the clinical importance because a good statistical significance does not always reflect into a clinical significance. So if we are looking at a reduction in pain, if we are not seeing, if we are seeing the clinical significance, but we're not seeing the statistical, we may be okay with that. But if we are seeing significant statistical findings, but we are not seeing it in the clinical aspect, it's not a drug that we're going to push for approval. I don't know if that's clear. If someone don't want me to take it again, I can do that. But that's what we discussed for my group. Thank you.
Thank you very much indeed. We have got 5 more minutes, and I would really welcome any other additional point for other members of the group or any questions from the floor. So while you're thinking, I would like to invite Dr. Kennedy to elaborate on that. Would you do that, please?
Yeah, thank you. Just to support what Jessica said, I think what is important for regulatory authorities is it's one thing to look at clinical significance, it's another thing to look at statistical significance, but more importantly, once you have the knowledge of what is clinical significance based on the dossier in front of you and what is statistical significance, where your skill and ability becomes crucial is balancing that evidence to make a final decision. And make that decision knowing that you're making that decision, having thought through this carefully, noting that safety should be behind your back as you're making that decision. It's so important to assess and conduct a risk-benefit analysis of the decisions you're making. And this is primarily the reason why we have regulatory authorities in place, because they are there to make sure that whatever products or devices are put in the market are there not to cause us harm, but to give us the benefit that we need to improve our quality of life. So I'm glad with the conversations that have happened. I'm glad with the questions that people have raised. And all I want to mention is that you don't have to have the statistical, technical knowledge, know-how. You just need to understand the basic fundamentals around what to make as a call and balance your decision. Thank you so much all.
Thank you very much,
Can
Kennedy.
we also get the feedback from the other groups?
Yes, group 3, yes.
And from our group would be 6, so that would be in the other group down.
Do we have a rapporteur?
Yes, yes, please. So
Prudence.
from our group, first we define statistically the difference in the context that it aims to determine if there's any observed effect or difference in a study and whether a study occurred by chance alone. And it's determined by the p-value, which is less than 0.5%. So for statistically significant difference to be less than the p-value, it means there's less than 5% of the results to be by random choice. So for a clinical significant difference, it refers to the practical importance or the impact on treatment effects or outcome. Well, we can assess it. We can assess if treatment has a meaningful impact on the patient. We gave an example to say a drug may show a significant an SS reduction in the blood pressure by 1 milligrams, but it may not be clinically meaningful. So statistical significance and clinical significance must balance. On why would it matter? For regulators, it helps in decision making and helps us in coming up with guidelines. In summary, it matters because it helps reliability and practical value of treatment outcomes. Statistical significance tells us that an effect is real. It helps in making a well-informed decision in regulatory decisions and prioritizing the patient well-being. The second question, the challenges that are faced by the regulators, we summed up to say it's the balancing between a drug that is either clinically significant or statistically significant, and as regulators, we have to make a decision on a product approval based on the clinical significance or statistically significant. The objective analysis, where we have to remove the biasness, reproducibility, findings are significant. Have they met the standards set by the regulatory body to approve new treatments? I think basically that's what we captured.
Great. Thank you very much indeed. Any members of the group want to add to what Prudence has presented? Kennedy, I just wanted to ask you if you could please, because one thing that we hadn't talked about during the lecture is the multiple p-values, for testing the same hypothesis, and how do we deal with that in our decision making as regulatory assessors?
So there's so when you have multiple p-values, look at the assessment that was done on the multiple p-values, and ideally you want, if you have several groups, to ensure that multiple comparison tests was conducted as well to adjust for variations. What normally happens, everyone, is that when you have multiple p-values, because you're testing so many different hypotheses, there is a risk of bias, or there is a risk of inflating some of the findings that you get. So to avoid that bias, there's a lot of work that's been done to show that. So to minimize that bias, you are supposed then to do what is called multiple comparison testing. And there's a couple of methods. The most common is normally the Holmes method, HOLMS method, but there's a couple of other methods out there. So that is something you'd also want to look at when a dossier is presented with multiple p-values, whether they've adjusted for the several different hypotheses that are being tested. Thanks.
Thanks very much. Any questions? Any members of Group 3 want to add anything? Because we still have to move to Group 4. Now, before I do that, I just wanted to ask, William, are you with us? William?
Yes, I'm here.
Yes, I just want to see whether if you have anything to add, please.
Oh, no, not at all. I agree with the explanation that was given for the material that I covered. So at the moment, I have nothing to add. Thanks.
Lovely. Thank you very much indeed. So can we move to Group 4, please?
Good afternoon. So Group 4, on question 1, that's the even numbered rooms, discuss why reproducibility of trial results is critical for regulatory approvals. So we discussed that this ensures that robust methods were used and that there were no errors in the study. The results were interpreted correctly, and that the obtained results were not by chance. So it demonstrates that the ability to produce the same results. And on the second part of the question, where it says when reproducibility fails, what are the implications? So one of the implications would be that the applicant would have to probably redo the clinical trial, because would say it's not validated. It's not the results may not be reliable. So the second part of the question, which talks about describe how regulatory authorities evaluate if statistically significant findings are also clinically important. I think this one, the other groups had stated what we were discussing. There is statistical significance does not always result into clinical significance. The example was given, like in the other group, of a reduction in pressure, which might not be clinically significant when it comes to cardiovascular outcomes. And yes, I think that's what we discussed. Unless there's any other member of the group that wants to add, in case I've left out something.
Yes, please. If anybody, a member of the group, wants to add anything or any questions, observation from the audience. One thing that I just like to emphasize that, and I hope that Dr. Kennedy will agree, is that the way that these questions or topic been set is that assuming that the methodology of the study is sound, and also the statistical tests, or in other words, the statistical analysis policy or that they have applied, it's also sound. That has been taken. So before you get to this point, of course, those are the things that you have to establish as the assessor anyway. Would you agree, Kennedy?
Yes, there are multiple layers of factors that one needs to look at to have a well-done study, and what you have raised is equally important. Otherwise, you may just think about those significant results, whether stats or clinical, forgetting that how the study was designed plays a massive role. How you took care of confounding plays a massive role. So all these things need to be put in the context. Thank you.
One question that I have from group 4 is that if you don't have a statistical significance, but you have clinical significance, would you approve that drug?
I think it would depend. I think it would depend. And this is where, you know, many other players would come in. I mean, of course, in terms of the hypothesis, it's not statistically significant. So theoretically, it stands a low chance of, you know, being supported for approval. But there has been evidence in some of the work that's been done out here that while there was no statistically significant result, but some of the results that have been presented in a comparison between intervention and control do show that there's actually some benefit on the intervention. It's just that that benefit hasn't reached clinical significance. And if it is thought that, look, that benefit is going to reduce or have a good effect on patients, and so you may as well just go with it. But I have personally not participated in that. But what I always do is I tend to encourage the publication of results that are also seen as not being positive.
Yes.
Because this is where this conversation comes up as well.
Thank you very much indeed. On a light-hearted note, Kennedy, do you agree that the 5% level of significance is arbitrary, and that was based on Sir Ronald Fisher, that one day he was lying in a bath and 5 toes was sticking out of the water, and that's how he came up with the 5% significant level?
It's a hot topic, Sam. But because I'm a statistician, I disagree with you in principle. But I have been in meetings where that conversation has come up, and you know, it was a hot discussion between clinicians and statisticians. Yeah.
Anyway, just wanted to lighten the mood. Yes.
Yes, I hate to come in there. Perhaps we can just wrap up. I think the next 2 groups, one has indicated that the other groups have covered what they were going to say. So we can skip the groups. And I believe Kennedy has given the summary of how he feels about the reporting.
So, Maps, would you like to bring the session to close? We still have got the wrap-up session.
Yes, that's correct.
Hand it over to you.
Thank you very much. So just to remind everyone that you are all familiar with the course outline or the course timeline. Next week, we are having lesson 11, which will be catered by Peter Stonio, Assem, and Doug. And the topic will be clinical data safety, risk management, and mitigation. So interesting issues coming by. And I'm going to urge the students to just use the email if you have any additional information, any questions that you would like to post, or you can use the Moodle and write your questions about this. And to that, I would like to thank everybody, and we apologize for going 6 minutes beyond. Thank you for the interactions. Thank you very much. Goodbye.
Thank you very much, Map. Goodbye.
Thank you.
Yes.
Thanks to all.
Bye-bye.
So, Sam, would you send the innocent man to jail, or would you free the guilty man?
I would definitely do not free the guilty man.
Cheers,
All right.
Prof. Goodbye.
Bye-bye, Map.